As Dr. DiPeri just said, he challenged you to learn something unexpected, and hopefully in the next 30 minutes I will provide you with that opportunity. And I'd like to mention that that's probably an indication of my whole career as learning things unexpected, and that's been one of the things that I found has been great opportunity. As Terry mentioned, I started off as an engineer, a control systems engineer, and actually then got exposed to applying control systems engineering to dosing drugs for optimal therapy. And then clinical pharmacology just naturally evolved from that. And so what I'm going to talk to you today is just another extension of that that's occurred to me in the last few months at the Gates Foundation, working in the area of Zika. And actually the whole story started off with dengue, and a project that we began 11 years ago in something called the Grand Challenges Exploration. So the Gates Foundation funds an effort every year called Grand Challenges, where we try to fund, for lack of a better term, crazy ideas that no one else would fund that might have some interesting opportunity to have impact. And this was one of those crazy ideas that we started 10 years ago to look at ways that we could perhaps interrupt the transmission of dengue. And it's evolved very steadily with great scientific progress over the last 10 years. And then in the last 6 months or so, Zika came on board, and with Zika hitting the world in a much larger way than it had in the past, it's really caused a huge acceleration of some of these efforts. So I'll give you a little bit of background on this program and try to relate it to what you're all familiar with in terms of clinical pharmacology. You see the logos from the Eliminate Dengue program, and so this is actually work that's done all by our partners, led by Scott O'Neill at Monash University in Melbourne, and a team of collaborators that are now throughout the world where there is dengue. So let's start off with just talking about what is Wolbachia. So Wolbachia it's a naturally occurring bacteria. It's inside of 60% of all insect species, and it's in all likelihood, if you ride your bicycle like I ride my bicycle, it's in probably a lot of humans as well, because I end up catching bugs in my mouth all the time as I'm riding my bike. And so it's a natural transmitter to humans, although it doesn't seem to stay resident in humans. And in insects, it's gotten around in insects through this very interesting transmission pattern called cytoplasmic incompatibility. So if we have a female here that's showing it. So there's our female who's infected with Wolbachia, and she mates with a Wolbachia-infected male. Then all the eggs in the clutch will have Wolbachia. If an infected female mates with an uninfected male, same condition. But if a male with Wolbachia mates with an uninfected female, then the hatch is sterile. And so in this way, this vertical transmission is the way the Wolbachia propagates through a population. But it doesn't transmit horizontally, so it's not dramatically infectious. It's probably brought across insect species over a long, tens of millions of years by other parasites that would take it from one species to another. And there's a suspicion that there's a particular wasp that's involved in maybe transmitting it across different insect species. But normally, if you have Wolbachia in insects, in a mosquito, it won't show up, for instance, in honeybees or in butterflies, though some of them do naturally carry it. So it lives inside the insect cell, and it's transmitted through the eggs, and it can't be transmitted to the warm-blooded animals. So therefore, this is viewed as an approach to look at control of the vector that transmits dengue as something that might be safe for humans and animals and the environment. And it's not a genetic modification. It oftentimes gets bundled, most recently in the last 6 months, in articles with genetically modified mosquitoes and sterile male release techniques that have been proposed for controlling Aedes aegypti for these diseases. It's really a biological replacement strategy for Aedes aegypti. And the key is you have to introgress the Wolbachia into the mosquito itself, and then allow it to start to lay eggs that have the bacteria present in it and begin to build up in the community. And when that happens, you get what I call here the Wolbachia dose-response curve. So if we introduce Wolbachia producing mosquitoes into a natural population, then you'll see this progression that occurs over time where eventually the whole population is carrying Wolbachia. It comes at a small fitness cost to the mosquito, but seems to not be substantially large enough to put them at a disadvantage for other invasive mosquito species. And it's not naturally occurring in Aedes aegypti, though interestingly it does occur in Aedes albopictus. but at a fairly low level. So when we establish it at a fairly diffuse level throughout Aedes aegypti, it begins to progress, and over a number of weeks, here you see in this cartoon a release of mosquitoes over 12 weeks of time, it gets to a critical threshold of about excuse me, 50 to 60% of the population, whereby then it spreads. And now you have Wolbachia that's contained within the population. And when that occurs, this interesting result happens. So this is some lab data that was in a publication by Neil Ferguson in Science Translational Medicine last year. And what you see are each line is a patient from a hospital in Ho Chi Minh City who had dengue. And that patient blood sample was taken from that patient, and then that was fed to an Aedes aegypti mosquito. And the mosquitoes that were wild type, that had no Wolbachia, are shown always in these columns to the left of the color chart here. It's a little hard to move over there on the green, but the left sides. And the ones on the right are the mosquitoes that are infected with the Wolbachia species that's called W. mel. And you may recall I mentioned that the researchers are from Melbourne, but W. mel is actually mel for melanogaster. So it's a species of Wolbachia that comes out of the fruit fly. And it's present among us and across nature in a large way. And so you see the little blue dot lines here are the viremic load of the patients. So they're increasing as you go down to the bottom of the chart. And the greens are the ones that basically showed no virus in the mosquito after feeding on a virally infected blood meal, and the reds are the ones that do, and the yellows have some moderate level. And so you can right away visually see there's a difference between the W. mel and the wild type. That was for dengue 1 strain, and then for dengue 2, 3, and 4, even more green that's present. And so Wolbachia in this in vitro or in vivo laboratory experiment essentially was showing a strong amount of blocking potential of the virus for all 4 strains of dengue. And then when you feed this into a mathematical model that then looks at the transmission of the virus through a population and assess the impact on the R0, so that's the transmission parameter from the standard epidemiological transmission models, you see that you get near a 70% reduction of dengue 1 transmission and upwards of 80% for dengue 2 through 4. And so this estimate's likely to be conservative, but this is a strong blocking potential and really should have a big impact on the ability to prevent dengue outbreaks from occurring in a region where Wolbachia is present. And so then here are some actual data from field studies that have been done in Cairns and Townsville, Australia, which is in the northeast corner of Australia. So they don't normally have much dengue in those areas, but they do get dengue when Australians come back from Bali on holiday and bring dengue with them because they have the vector in the right climate for dengue transmission, and so they get outbreaks that occur. And so here you see the green hatched areas when the mosquitoes were released and a pretty rapid uptake that then has been sustained. And this has been sustained now for 5 years. And one of the things I'll note in the little cartoon, if you look just after 2014, there's a bit of a cyclone there. So there was a cyclone that came ripping through the neighborhood, and the population stayed sustained. So it seems that it's actually been relatively stable and been able to hold fairly strongly in this part of Northeast Australia without too much problem at all. And since then, there's been no locally acquired cases of dengue that have been demonstrated, all transported in, and no outbreaks that have occurred as a consequence of those in the regions where they have Wolbachia released. So right now, the project has been releasing mosquitoes for the last 5 years. Importantly, one of the first places that the investigators went was their home country and their home neighborhood. So they didn't go off to some other country to try this out as an experiment. They actually did it first in Australia, built a body of evidence in Australia, and then moved on to other places in the world. So here's a map of one neighborhood in Cairns called Parramatta Park. And the map on the left shows dengue cases over, I think it's about a 13-year period within this one area of Parramatta Park in a 1 kilometer radius around it. And you see the degree of concentration that's occurring. And then the Wolbachia were released in the Parramatta Park area, and since May of 2013, this has been the number of cases. And one of the things just numerically that they looked at is that if you take all the cases in that 1 kilometer circle that's drawn around the neighborhood, 34% of them were in Parramatta Park in the previous time before release of Wolbachia, and now since then there's only been 4. And the 3 cases that have actually shown up there all occurred at different times of the year, so they're not in any way related or correlated spatially or temporally. And so it seems to be that this is blocking the ability of even a transport or an imported infection to be able to be transmitted through the area. And then here are some of the results as well, similarly in other field sites. So the upper curve is for Townsville, Australia, and you'll see all the red bars show the cases of dengue that existed before release. The green hatched area is when Wolbachia is released, and you start to see a drop down in terms of the number of cases. The lower set of graphs to the left are for a region in Yogyakarta, Indonesia, and that's a place where I'll talk about in a minute. We're doing a pivotal clinical trial coming forward. And again, you see some still degree of Wolbachia cases that occur, or dengue cases that occur in the Wolbachia release. And then the other one to the lower right is actually in the area of Nha Trang, Vietnam. And there the releases happened in this small island called Trinagan off of the coast of Nha Trang. And since they've had uptake in Trinagan, the only case that's been reported has been a single imported case that's occurred. So now they've gone 2 years with no dengue whatsoever. But notice the degree of dengue that's occurred just across the inlet from the island. So it's present completely around them, but it's not showing up in the mosquitoes in their own population. Now, one of the advantages of this is, and a challenge as well, is that the Wolbachia don't spread diffusely throughout a region, right? Partly that's because Aedes aegypti don't spread very far. They're actually fat pigs. They like to just stay put wherever they're present, and they have access to a blood meal, and they don't travel very far. And so as a consequence, they don't take Wolbachia outside of the places where you release it. But the good news is once you establish it in a region, you also don't get back dilution in from other parts in the neighborhood. So it actually helps. And so that's one of the reasons why this island has been essentially this little spot that we've been able to claim as essentially been dengue-free and is holding over some period of time. So then this led the investigators to ask the question, well, if it blocks dengue, what might it do to some of the other viruses that are transmitted by Aedes aegypti? And so a year and a half ago, they took the strain of Zika that had been first found in Africa, in Zaire, then in the late 1940s, and got the strain from ATCC and purposely infected mosquitoes with blood that had the virus in it, and then did assays at different times, 6, 9, and 13 days after feeding. And they looked at the presence of virus in the midgut of the mosquitoes as well as in the saliva. So the key thing to be able to see, is it being passed to the point where the mosquito itself could transmit it to a human? And here are the results that are summarized both from that study and then 2 recent studies that have been done by our collaborators in Colombia and in Brazil with the local strains of Zika that are currently circulating. And one of the things you can see here is these are midgut infections, so the black bars are always the wild-type mosquitoes, the green are the Wolbachia infected, and you see over time that the infection rate drops down in the midgut. You would expect to see infection in the midgut because I just took in an infected meal, so that's where it's sitting. But notice, importantly, there's very little infected virus that's in the salivary glands. And here from the results in Colombia, again, the same transmission type of experiment, very little in the midgut after 10 days, and none in saliva and none in the disseminated throughout the body itself. And then these most recently published in Cell by Luciano Moreira from Fiocruz in Brazil, showing again similar results and the ability then to take virus that was from the mosquitoes that were infected, the wild-type mosquitoes, and see if you could transmit them back in to cause the infection to transmit forward. They showed again that mosquitoes that have W-mel in them as a Wolbachia strain basically block this transmission. And so it appears that the ability to block Zika is even stronger than it is for dengue. And then they repeated this again with 3 different strains of chikungunya, and they saw exactly the same results. So there seems to be a strong ability of this bacteria to block the transmission of viruses, of flaviviruses at least, through Aedes aegypti. And they haven't tested yellow fever yet, but in all likelihood it'll block yellow fever as well. So we have the potential now with a single intervention to essentially limit the transmission of 4 diseases transmitted by 1 vector, which is quite an interesting amount of potential. And the mechanism of how this works, we don't really know. But the hypothesis most recently sort of considered is that in all likelihood the bacteria are competing with the virus for cholesterol, and they're not allowing the virus to essentially replicate because they're just outcompeting it overall for cholesterol. And so that's one of the hypotheses that's being tested. to see if we could really understand this from a mechanistic standpoint. But nonetheless, it appears to be quite effective at blocking all these diseases. So then how challenging is it to grow these mosquitoes and to get them dispersed within a community? It's actually relatively simple. So here you see some pictures from the site in Yogyakarta, Indonesia. Egg strips are produced by essentially rearing mosquitoes in mosquito dorms. I don't know if you've ever seen them. They're called bug dorms, basically small square boxes that have about 1,000 mosquitoes in them. You lay felt strips in the bottom, and the mosquitoes lay their eggs on the felt strips. You desiccate the felt strips. You put them in a Ziploc bag with 4 pellets of fish food, and you put them in a Mozzie box. And a Mozzie box is basically a Chinese food takeout box. It's exactly what it looks like. You put the water in with the eggs and the fish food. You fill the box halfway with water, and you put it in a shady spot in your yard and close it up and walk away. And so essentially what you do is you release the good mosquitoes, and over time they start to replace the bad mosquitoes. And depending on the amount of mosquitoes you have in your yard determines how many times we might have to redo this on a biweekly basis. But the important thing is that this is a community-focused event to take this on as an eradication strategy. The only way this works is by getting people in the communities where the disease is present to take this up and to work with you in collaboration. So currently the program sites are located throughout the world, and I've broken them up and colored them here by 3 different designations. So the research is still ongoing in Cairns and Townsville, and Melbourne is the center where a lot of the basic biological research is ongoing, as I said, led by Scott O'Neill and Cam Simmons. And then in the blue sites in Vietnam, Nha Trang, and Indonesia and Yogyakarta, we're about to begin a cluster randomized trials that would provide pivotal evidence that this is actually effective at blocking transmission of dengue. These were all efforts that were being planned in 2015. The Yogyakarta trial will begin just after the end of Ramadan, which we just started yesterday, and the Nha Trang trial will probably begin in 2017 and read out in about 2019. And then with the onset of Zika and its explosive release across the Americas, there's been a great interest in Colombia and in Brazil to be able to actually do a massive scale up. So in the 2 places where we're operating in those countries, we had small scale projects, and the governments and health ministries of both of those countries have approached us to do a much larger scale release. And so in fact, what we're going to do is a large scale deployment and learn as we do large-scale deployment. Because one of the things we need to figure out is what is the optimal dose, what are the characteristics that add variability to the dose of mosquitoes that one has to use to be able to get a stable Wolbachia presence within each of the communities. And so in Townsville, Australia, it looks like this. It's a beautiful place if you've never been. It's relatively not built up, so small size city. There we've been doing large-scale deployments for the last 2.5 to 3 years, and part of the efforts there are exploring new ways to involve the community and develop low-cost and effective methods for delivering the technology. So far, to cover all of Townsville, it's a city of about 50,000 inhabitants and 15,000 houses. They've done 3,000 mosquito bucket releases that have basically accounted for about 1.3 million mosquitoes. Now, I want to give you some sense of that number. 1.3 million mosquitoes sounds like a lot of mosquitoes. That's over 2.5 years that they've been releasing those. If you're doing any of the sterile male release techniques that are being explored right now, they generally release 1.3 million mosquitoes per week. So that's a big difference in terms of the overall amount of biomass that needs to be released in this program. And importantly, with this program, once it gets established, it doesn't need to be reseeded. So once you actually have Wolbachia in a territory, it appears that basically it stays stable and you don't have to reintroduce it again and again. And that's not the case, again, for sterile males. So as a cost-effectiveness technique, this actually has a lot of potential to be quite cost-effective for reducing disease burden. In this case, and so far it's been US $16 per person for the deployment in Townsville, mostly because there's a lot of work going on in monitoring and research. And Townsville is not a densely populated area. When you get to a place like Rio de Janeiro, those costs go down dramatically because you're essentially releasing the same number of mosquitoes per square kilometer, but there's a lot more people that live in those square kilometers. And one of the things that they've been actually exploring is different engagement models. So here you see description of the Wolbachia Warriors program. So this has been a great citizen science program in Australia, and actually now transmitting transferring as well to the other places where we're working, where they've engaged essentially middle school children to be able to release mosquitoes in their own yard and keep track of the release. And so one of the things you can do is take the lid off of the bucket at different numbers of times. This is when they were using buckets before the Chinese food takeout box, and take a picture with your cell phone of the water to look at the larvae, and from that you can actually, there's a digital app that the lab has developed that can tell you what percentage of the larvae are alive and what percentage are dead. So students can take measurements on their own buckets, they can look at the release characteristics that occur over time, and then be involved in the science and understanding this project. And it's become very successful, and they're enrolling 4 more schools this year and another 2,000 Wolbachia Warriors that are coming forward in Australia. I mentioned Indonesia is a place where the pivotal trial is going forward in Yogyakarta City. So Yogyakarta City is a very different city compared to Townsville, Australia, much more densely populated, and the large-scale city release is going to begin. We've already been doing 4 releases in the outskirts of the city just as essentially demonstration projects and to build essentially collaboration and consensus about running the program with the population. And here you see the uptake for these places in Yogyakarta. Notice a wider number of the green zone, so a greater mosquito intensity or burden there, and so greater number of releases that occurred. And again, one of the things that Scott O'Neill likes to mention, he said he's a laboratory researcher, and so here you see this little volcano here. So about 3 months into the release, then Yogyakarta had a volcanic release that covered the whole city in about 6 inches of ash. And so that essentially stopped the releases. But notice that the Wolbachia didn't totally collapse. So they reestablished and continue to do the releases afterwards, and it's been sustained since then. And so this is going to be the place that they expand in a cluster randomized trial. And one of the challenges was to try to figure out the impact of this technology on disease burden. And the big issue is if you go to Yogyakarta now and measure, do serological surveys on people, you see just about everybody showing up positive with dengue at some point. All right, so it's already present, profusely throughout the population. And so one of the ways we're going to have to track this is to put in sentinel sites for febrile patients and then do assay to actually confirm it's dengue or not dengue, and then work back essentially in a cohort way to look at who lived in a Wolbachia established neighborhood versus who didn't live in a Wolbachia established neighborhood. Now the challenge here is that people of course can get infected with dengue not just where they live but where they work or on the bus station or somewhere else if they're all at a party at a beach or something like that. So it's not a perfect way to be able to assess the impact of the technology, but what we should see happen is that because it blocks the transmission, when someone is infectious it won't become explosive in their neighborhood. So we should ultimately see that the places that are covered with Wolbachia essentially are blocking these bigger outbreaks compared to the places that aren't. And what's happening is essentially they're starting in the northwest and the southeast corners of the city with releases and then moving forward towards the middle. And so it's in essence like a modified step wedge trial, because you'll always have a spot that is a control region that hasn't had Wolbachia release, and you can compare what the rates of dengue are during time, because dengue has a very cyclic pattern to it, and so it'll be important to be able to compare it to the places where Wolbachia is not established over the course of the whole study. And then, I don't know why, but for some reason, this unfortunate pink region was the place that's left alone as a control, so it won't be treated at all for the whole duration of the study, and that will essentially become a sentinel comparison site. The other places then where we're doing these large-scale releases are Colombia, so here in Medelin and the small area called Bello, which is just north of Medelin. And again, different kind of topography, different kind of houses. Here we've released in the Paris neighborhood of Bello, which is 32,000 individuals and 6,000 houses, and that's had a good establishment of Wolbachia over the last year, and now we're actually in the process of doing a rollout of all of Paris and then starting to work towards Bello and Colombia. And here you see actually the intensity maps of dengue in the 2 cities. And the red shows the regions where there's lots of dengue. The green is where there's very little. And we hadn't appreciated this when we were getting started with this project. But in fact, Medellin, Colombia, and Bello are quite interesting places to do this kind of study because they're sort of inland islands, right? They're valleys that are surrounded by this high plain. So there's no dengue and no transmission of disease around the perimeter of the city. So we have this interesting opportunity to walk in and essentially disperse throughout this valley Wolbachia Wolbachia mosquitoes and see if we can, over time, see the progression of the disease essentially go away. So working in strong partnership with the Ministry of Health, with the Mosquito Control Board there, we're adding this to their ongoing efforts of mosquito control. And this is going now from a city of 25,000 inhabitants, where we were working in the Paris neighborhood of Bello, to 2.5 million people for Medelin and another 500,000 for Bello. So 3 million people is going to be quite an interesting scale project. And our goal is to see if we can cover this whole territory in the next year. So this is what I do on my nights and weekends when I'm not doing my day job with the Gates Foundation. And then Bello, interestingly, has advocates and release coordinators. Again, this whole program is built on collaboration and cooperation with the population where the work is going on. And so each of the communities that we work takes this program and converts it into their own way of communicating it to their peers. And so here you see on the bottom the cartoon figures that were made in Colombia for describing how the technology works, the ability of the good mosquito to essentially drive out the bad mosquito. The children in the school put on a play and essentially made it into a telenovello about the mosquitoes mating and causing good mosquitoes that come out of that relationship. And so that's been quite interesting to see how they've developed that into their own ways. But you see the group on the left is actually a senior citizens group that's gone out and become the advocates. And so they're the ones that talk within the community. So if anyone in the community has concerns or worries about the program, they bring it to their own peers, and then that's filtered back to the scientific group. The group on the right it's a single moms group that's been really strong advocates for dispersing the mosquitoes and the information throughout their community. So it's been a nice empowerment effort in these places. And then the other place where we're doing a large scale up is Rio de Janeiro and Niteroi, Brazil. So Niteroi is the city to the east of Rio across the bay. Unfortunately, we will only have certain places covered during the Olympics. So if any of you are attending the Olympics, I would highly recommend that you seek housing in the Jurajuba neighborhood of Niteroi. It's not a tourist region, but it is a place that we have completely covered with Wolbachia. And so it's been interesting. So Rio, Medelin is 3 million people. Rio is 6 million people. If you add on Niteroi, it's 6.5, almost 7 million people. So quite a big task. And one of the things we're really interested to learn is, how do you take a program like that to scale, and how do you do it in an efficient way? What are the things that we can measure and model along that to teach us how to roll this technology out more effectively if we can show that it does have the impact we think it will have. And this was a real interesting experience for me. This is actually just showing you part of Niteroi in the neighborhoods that are broken up in it. And the place where we've been doing the releases are just down here in the end in this place called Jurajuba that I was mentioning. And Jurajuba is actually, it's a municipality that's made up of 6 barrios. And so the team had done releases in one of the 6 barrios and completely covered it. And it's a very favela-like structure, houses built on top of houses. The sentinel mosquito catches that we were doing in those neighborhoods were pulling in 250 Aedes aegypti in a tiny house a week. Most of the other places where we do these sentinel catches catch 25 Aedes a week. And so this place has an incredible amount of biting pressure that's present there. Nonetheless, the Wolbachia has been introduced over the course of about 6 months to get up to the appropriate level. And this picture here is essentially the community council that we were meeting with. And these ladies over here, they run the community health district in Jurajuba. And in Jurajuba, that district covers all 6 of the barrios. And one of the things that they said to us, they stood up and said, since you guys have left these mosquitoes in our neighborhood, and we have them established in this neighborhood in Jurajuba, we don't have any more dengue from that neighborhood. Whenever we get dengue, it comes from the other 5 neighborhoods, but it doesn't come from this one. Can you please come and cover the other 5? So we're in the process now of doing the other 5. They'll be well established by the Olympics, so that's why I say Jurajuba, that's a place to look for as a possibility. So then the other thing is, how do you get this out to the rest of the world where dengue and Zika and Chikungunya are big concerns? And so one of the things that the team has put together quite effectively is what they call a kit of parts. And this kit of parts is basically just the box of things that you need to do to establish your own program in the community where you work. And so this is a website called Catalyst, where they essentially retain the information provided as teaching and training for anyone who wants to start to build the opportunity to do Wolbachia releases in their town. They use it for transferring of knowledge and methods any kinds of procedures and standard operating procedures that go forward, protocols, templates, and troubleshooting guides, real-time access to program expertise and advice via an exchange, so it actually creates a dynamic environment for people to communicate, and importantly, native language support. So here you see the swing over to the Vietnamese site. So it's been developed in the 6 places where we're working right now. It's been a tremendous system for creating a community among the communities where they can share information with each other. And one of the fascinating things has been to see the population get really engaged and want to start to engage with other places around the world where this has been released. So I'll stop there and just leave you with our motto at the Gates Foundation that every person deserves a chance to live a healthy and productive life. That drives what we do. Everything I know about mosquitoes and Wolbachia I've learned in the last year and a half, and it's been a really fascinating ride. And as Terry said, we're starting to use a lot of the same principles in terms of dose, exposure, response, and modeling to understand how to effectively roll this program out. But we think this actually could be one that has big impact. And as I've said in the bottom of the last slide on Jurujuba, I think this is a kind of program where we won't need a statistician to say that it works. So it either works and the people know it, like the nurses who told us in the community, or it's not effective, and then we'll have to look at other approaches. But we have a hunch so far from what we've seen that this could be quite impactful. So I thank you for your time, and if there's time, I'll take a question or two.